Hello everyone. My name is Shomanetropal. I am a student of Electronics and Communication Engineering from St.Thomas College of Engineering and Technology. Today I will be presenting a review paper on application of biomems prepared by Saswata Das, Shoykot Dotto, Indrajit Dotto, Kirtibhushan Ganguly, Ujjwal Pandey, Shankhadeep Mondal and me under the guidance of Proshnatit Pal. In this paper, we aimed to illustrate the immense application of microelectromechanical systems in the field of biology and, to be precise, in the field of medicine. Miniaturizing is something that is aimed for long to make instruments fit in minute places. And with the advent of biomems, we finally managed to get certain instruments which are miniaturized and can be applied in the field of medicine. And that is what we are going to explain in this review paper. Biomems has been a boon in all sense. Besides miniaturizing an instrument, it also made it economic and more accurate to the job we will be doing with it. Moving on to the first step of creating a biomems sensor. To create, we follow regular fabrication techniques to create a wafer and finally get the sensor we are aiming to. Speaking about sensors, they can be classified into three broad categories, namely microchemical sensors that deals with environmental changes like pressure, electrochemical sensors that respond to chemicals, and optical sensors which respond to fluorescence or other plasmon resonances. And finally, we are up for the first application of biomems in the field of diagnostics. We use certain techniques like microarrays, PCR chips, and self-diagnostics to diagnose any disorder. Microarrays are arrays consisting of known DNAs over which we put our sample DNA and analyze the match between the known and the sample DNA to infer a result. We use PCR chips to amplify the content of a cell while extracting any component from a cell to analyze that we get it in a very small quantity, and analyzing that small quantity increases the chance of error. So what we do is we use PCR chips to amplify that component and then analyze it in a proper way. Moving on to self-diagnostics, these are instruments that help us analyze blood sample or urine sample or swab sample without following the regular laboratory procedures, and which makes it quicker for the treatment to start, which helps a patient to recover soon. The next application of biomems is in tissue engineering and cell culture. In this procedure, we target a cell, identify it with the process of cell sorting, remove the cell membrane to get into the inner components with a process called cell lysis, and then we extract that component of our interest with purification and amplify it with PCR chips, and finally analyze it to infer a result. Stem cells are another very interesting application of biomems, and especially using tissue engineering and cell culture, we can experiment on stem cells of different plants. We all know that stem cells conduct water and other minerals to the whole plant. Now, those stem cells could be made to transfer water and other minerals in lab. Also, we can create virtual environment so that those stem cells can reproduce in an artificial environment. And the most interesting fact in that part of creating an artificial environment is we can create an environment in such a way so that we can make those cells grow conditionally. Moving on to the next topic, medical implant and surgery application of biomems. This is one of the biggest application of biomems in the field of medicine. Nowadays, microsurgeries are done, and for that we need instruments which are of micro or nanoscale range. Instruments like scissors, needles, and scalpels are made of the desired size with the help of biomems. The next important application is in drug delivery, where we need to control the adequate flow of the drugs that is to be supplied to a particular point of disorder. Moreover, we use microreservoirs to hold the drugs and micro valves and pumps to make the adequate flow of the drug possible. Here comes the next topic of prosthesis. I was talking about implanted sensors under the sensor section of biomems. Using those sensors, we can replace a part of a nerve cell that is malfunctioning, and that type of sensors are called regenerative electrodes. Now, the process in which we replace the part of the malfunctioning nerve cell with a regenerative electrode is called prosthesis. So far so good, we were talking about miniaturization of instruments. The implanted sensors, which are also miniaturized with the help of biomems, can often be considered as a foreign body, and our body can react to it and start blood clotting. And if blood gets clot, our objective is contradicted. So what we do is we create a virtual environment so that our body does not feel that the generative electrodes or the implanted sensors are actually foreign materials. To create that virtual environment, we needed some kind of fluids called microfluids. Those microfluids are also stored in a micro reservoir and pumped according to the need in an adequate amount to the point of delivery. These were the major applications of biomems. With the discovery of pregnancy test to today's place of biomems in the field of medicine is remarkable. With the present ongoing research works, we can hope to achieve great standards of treatment. We finally want to thank Society of Technologically Advanced Materials of India and Assam University, Silchar for accepting our project and allowing us to present on this. Thank you.